All right, ladies and gentlemen, let's review the structure of DNA before we get too much further into how it fits in the nucleus, how is it even replicated, and then furthermore, how is it taken from DNA to RNA to protein. So, the two sides of the ladder, right, this part here and this part here, are comprised of alternating sugar-phosphate bonds. Let's get all this stuff on the screen. There we go. Of alternating sugar-phosphate molecules and bonds. The rungs across the middle here, right, which are held together by hydrogen bonds, are comprised of the nitrogen bases. Two bases make each section, each rung of the ladder, right? So one base here, one base here, one base there, one base there. So two bases connect across the middle. And obviously, we know they're held together by hydrogen bonds at this point. Adenine always pairs with thymine. Two hydrogen bonds form between adenine and thymine. Guanine always pairs with cytosine, and three hydrogen bonds form between them. We don't need to know the number of hydrogen bonds. That's just a little added tidbit of information. I really just care that you know the base pairing rules. A pairs with T, C pairs with G. Now, DNA is a carrier of information. Let's kind of speed through this. So DNA must be able to do a couple things. It has to be able to carry information, and it must be able to do this well, do this efficiently. The information is carried in the sequence of bases. So a sequence of bases, for instance, if we look at one side of this DNA molecule, T, G, G, T, T, G, G, T, that's a sequence of bases, right? If we reverse some letters or we put an A in or a C in, that's going to be a different gene. That's going to be a different sequence. That's going to be a different phenotype. So since the number of paired bases ranges from about 5,000 to the simplest virus to 6 billion in human chromosomes, variations in these base sequences are seemingly infinite. And also, I have to double check my numbers here. I don't think 5,000 is the simplest virus. I actually think it's much less than that. All right, and that bottom part on that slide did not matter, so don't worry about that. All right. So how do we fit 6 feet worth of DNA, 6 feet worth of a molecule into the submicroscopic space that is the nucleus of a eukaryotic cell? Well, the structure of the chromosome allows the DNA to be packaged tightly inside the cell. So it's this actual structure of the chromosome. So the chromosome, we know a chromosome is actually comprised of DNA and proteins, right? It's not just DNA anymore for us. We know that there's protein involved as well. So the DNA, right, the nucleic acid, is actually wrapped tightly around certain proteins, and we call these proteins histone proteins. Okay, as you can see pictured here as these little pink balls, these are histone proteins, and the blue strand-like structure is DNA, right? Being able to wrap around these small proteins allows a lot of DNA to fit inside of a tight package like the nucleus. And together, once the DNA and the histone molecules form a bead-like string, or a bead on a string-like structure like we see here in this image, we call those structures with DNA wrapped around the histone protein, we call it a nucleosome. Okay, so first on the far right, we have the DNA double helix. Then, down here, as we move further back toward our chromosome, we have histone proteins, right? These are the proteins that DNA wraps around to fit into that nice tight package that is a chromosome that fits nicely inside the nucleus. And then we have protein, these histone proteins, wrapped around some DNA, or DNA wrapped around them, and we call that a nucleosome. And then those nucleosomes coil up on each other. And then we get what's called supercoiling, which is the coils coiling further, until we ultimately get the chromosome. Now, nucleosomes seem to be able to fold enormous lengths of DNA into tiny spaces available inside the cell. So it's this formation of the nucleosome, the wrapping of DNA around these histone proteins, that allows DNA to form the chromosome structure and fit within the eukaryotic nuclei. Okay, so now before we talk about how DNA goes from a molecule to a physical trait, a phenotype, we need to talk about how DNA is replicated, right? Because DNA is replicated so often in our cells because our cells are so often going through mitosis and or meiosis. So the structure of DNA actually allows it to be copied easily, as you can kind of hopefully tell in this illustration to the left. So another word for copy in this sense is going to be replicate. All right, so DNA is able to be copied or replicated easily. So each strand of the double helix, each side of it has all the information needed to construct the other half, right? Thanks to complementary base pairing. If you split the DNA molecule down the middle, if you have a C on this side and you take away its G, to build a new strand, you have to add another G. So that C codes for a G, okay? A would code for a T. T would code for an A. So each side has the code to make the other side. And because each side can be used to make the other strands, it is said to be complementary. Hence the term complementary base pairs, A pairing with T, C pairing with G. So the strands are ultimately separated, and then the rules of base pairing is what allows new strands to be made. So if we look here, we see that this initial strand has been separated or is being separated. And as it's being separated, look, a new strand is being added to one side. Another new strand is being added to the other, following complementary base pair rules and making an identical DNA strand to this one. And here's how we define replication. Excuse me, folks. It's the process in which a DNA molecule builds an exact duplicate of itself. Simple as that. Okay, so if we draw replication out real simply, we're going to learn it to a little bit more complex of a degree, but this is for our baseline knowledge. What I really just, if you leave here today with nothing, I want you to leave here with this. All right, baseline. So the parent molecule or the DNA molecule in panel 1 has two complementary strands of DNA. Here is one and here is the other. Each base is paired to its specific partner by hydrogen bonds. Adenine pairs with, that's right, thymine, and cytosine always pairs with, yep, you got it, guanine. So the first step is the separation, which we see happening here in panel 2, of the two DNA strands. The hydrogen bonds are basically broken. That's how the separation occurs. And now each parental strand serves as a template strand for an entirely new DNA molecule. So we had one DNA molecule, we separated it, and now in panel 3 you see we're going to use each separate strand to build a new strand ending with two new DNA molecules, or technically an additional DNA molecule. So new nucleotides are inserted along both sides, both templates, as the molecule is being unzipped, and we'll talk about that in particular here in a few slides. And nucleotides are added one at a time following base pairing rules. A pairs with T, C pairs with G. And just like before, the hydrogen bonds naturally form between the complementary base pairs, and the DNA molecule begins to retake shape once again. All right, folks, I'll catch you in the next one.